So Dayanne and I are going to switch it up, and I'm going to give you guys all the sort of fluffy intro. I'll talk about methods and show equations. Don't worry. But Dayanne has a new result that's very specific. You know, it's a single paper, and so we thought instead of doing something very focused followed by an application, we'll do the application first, get everyone very comfortable with the basic ideas, the core methods, some of the older methods, and then Dayanne will show you the cutting edge after that. And I think it'll work in terms of not repeating the introduction and things like that. So that's the main reason for this. We do a lot of different things in the lab, and a lot of what we do is really focused on inference of networks. Some of our papers are about ecological networks, some on social networks, and we're going to focus today on biological regulatory network inference, and the focus will be even further on things involving transcription and chromatin structure. If I get a chance, I'll describe some of the work we do with ecological networks and fitness networks, just to sort of really highlight that network inference is not a single thing, that often things that look like the same problem are not for various intense reasons. And so with ecological networks, the methods are different, but you see in the literature people using the same methods incorrectly, and I want to maybe save some time to point that out. Okay, and again, this is what I'll do. One of our favorite little guys in the laboratory is B. subtilis. We love to develop methods and see if we're out to lunch with B. subtilis and E. coli and things like that, but we like B. subtilis more than E. coli, just because. I'll go through a few of the things that we do, and I'll introduce what's now maybe a bit out of date, but at least it establishes sort of the core model. Then we'll go into some of the current things, and then Dayanne will take it home with how we apply that to multiple data sets and multitask learning. I'm going to skip over some of these things, one because Dayanne's going to talk about it, and one because it's still not quite ready, but I'll show you a little hint of some of those things as well. Okay, and we do tend to skip from B. subtilis. We fly over yeast, we skip mouse, and we go straight to human. And so that's a little weird, but it kind of works. All right, so regulatory networks, since it is early, we'll just start a little fluffy. So regulatory networks are not all that there is, but regulatory networks are very famous for establishing fate decisions. And much of what we see in the single-cell RNA-seq world, much of what we see in the network inference world is really aimed at fate. And how do we establish fate as cells divide and become terminally differentiated in different ways? And that's one of the things that I'll build up to today. And that's one of the main reasons why we do regulatory network inference in the lab, is to look at fate decisions in rice, fate decisions in bacteria, and fate decisions in mostly the immune system. I think more and more now, as we explore the immune system, we realize that function can establish alternate programs that are expressed in the chromosome and in the transcriptional regulatory state. And so really what we're mostly interested in going from just a picture where you grow cells and you do experiments and you really focus on fate. We're really thinking as hard as we can about function, and beyond knocking genes out, but actually function with reasonably interesting different perturbations. For the immune system, the interesting perturbations tend to be what's happening in the microbiome, what's happening with pathogens. They tend to be how those various stimuli are integrated with the genotype of the individual, which is also variable. And so fate and function and genotype together, a great way of approaching that very complex system is through understanding the regulatory network. From a more, and this is, again, since I think there is a mix of backgrounds, I'm going to show you the end goal, and between Dayanne's talk and my talk, we're going to barely get there in terms of how to make models this big. But one of the things that we have to think about is that regulatory networks, even if we focus on a small set of regulators, and this is 5 regulators that are verified and known to be extremely important in the differentiation and the function of T helper 17 cells. These are cells that are involved in inflammation. They're found in the lamina propria, the space right behind your gut. They're found circulating. They're found at lesions in all sorts of autoimmune diseases. So they're very important cells. They also have innate lymphoid cells, so they have versions that lack the T cell receptor. So they also have interesting cousins elsewhere on the fate map. So they're interesting for fate and function. If we look at just a handful of genes that genetics essentially told us were important, when we actually build these models, we find that there's strong support from many different experimental sources for these things actually having very large sets of targets. So this is a long-winded way of saying one of the key challenges with regulatory networks is that they're very large and that they are not going to reduce in the way that the mechanistic models you see at the ends of papers in Cell reduce, right? We're not going to be able to write down a cartoon that we can memorize and say that that's how the cell equilibrates and decides the level of expression of hundreds of thousands of loci. It's just not going to happen. Another thing that that's interesting is that many of these things, actually two of the guys on this slide, actually every single thing on this slide is part of a large family. And a large protein family that radiates recently in a lineage like mammals is telling us that there is some selective pressure to have all those bits and pieces. So biologists want to reduce things down, but evolution is telling us that actually we do need thousands of different regulators and they're all going to be simultaneously important. If that wasn't the case, then we might not see these information processing genes radiate like they have. And when I say radiate, I mean there's a homolog in yeast, and for some reason we've got 400 of them. You know, that's what I mean, evolutionary radiation, in case there's physicists in the audience. Okay, another last little point, and again, this isn't methods, this is a bit fluffy, but these are what we're trying to do is justify why we need automated methods and why we need methods that scale to very large sizes. Because for those of you who do modeling, you'll see that some of the methods we use are very weeny. They're very stripped down so that they can scale. So I'm justifying the scale is what I'm doing for people who don't like biology in the audience. All right, so when we look at what these guys regulate, it's a little too early. So I'll just we'll get rid of all the science and just do comedy routine. And then when you guys are awake in an hour, Dayanne will take the stage. All right, when we look at what these guys do, another reason why we need unbiased methods that scale is that the things that are important for fate and function are not the things we expect most of the time. The cell, for example, this was T helper 17. This is ancient history. This is more than 2 years old. We knew that cytokines and cell-cell signaling was a major way that these cells function. We knew that whatever this thing's doing, it's going to be tinkering with transcription factors to solidify its decisions, to canalize its functions, if you will. What we didn't realize is that apoptosis and stress, carbon metabolism, and a number of other things were really important. And when we thought about it, and actually the way that we first found this is there was another student in the lab working on cancer. They sent me the network file for this. I thought I was looking at the cancer network. I was looking at this network, and I got really excited about all these things. One of the key things we got excited about was an interaction between HIF1 alpha, which responds to oxygen stress, and redox stress. There was an interaction between that and basic carbon metabolism. And one of the things that we saw was that hexokinase 2 was upregulated and hexokinase 1 was downregulated. Now, hexokinase 1 is inhibited by glucose. So it eats a little bit and then it stops. And hexokinase 2 will chow down forever. It doesn't have this feedback, this negative feedback. So if you're going to be a cancer cell, hexokinase 2 should be high. And this is something that people know. Well, why should Th17 have this gene up? Well, when you polarize a Th17 cell, it divides and divides and divides as fast as it can. And what we found is there were lots of overlaps between the phenotypes, the molecular phenotypes in cancer in this cell, and the careful regulation of those was very important and very screwed up in some of our mutants. So this is a long-winded way of saying that only 3 of these groups of functional genes were thought to be important by the immunologists, and the network let us dig into some of these other areas and realize that the careful intercoordination of all these processes was essential. So there are many phenotypes, for example, that are inflammation phenotypes now that are condition-specific or cell-type-specific knockouts of these otherwise very boring housekeeping genes. Okay. one last slide for setup. Now, if we're going to use methods to learn networks from data, we could use the most stripped down methods. We could use linear methods. We could use correlation, mutual information. And actually, the early days of network inference, and actually maybe the current state of the art, depending on who you talk to, in network inference, is to actually do things that have no model. Meaning you take your expression values for your regulators, your expression values for your targets, you compute mutual information, you normalize it in very careful ways. Maybe you do this over multiple data sets and you stack the result. But in any case, you don't have time, you don't have concentration, you don't have activity, you don't have any biophysical parameters. The flip side is to model a regulatory subcircuit that you know very well and look at the regulators and the kinases and all the various things that modulate activity. And you build a very careful dynamic model. And so what we want to argue, or what I like to argue, even though I don't have, we haven't finished proving that this will work, except in a few cases, is that actually making a more biophysical model is something that we can scale to these very large regulatory networks. And in the end, one thing to remember is that when we're, if we start putting time and concentration and the activity of regulators in as explicit variables, even if they're latent, we increase the complexity of the model quite a bit. If we start to model interactions between genes, we also start to increase the complexity of the model tremendously. But what are the actual advantages of that? So if we try to make biophysical models, we're making our life much more difficult, and there are many, many general machine learning methods that we can't use anymore. Or we at least have to heavily modify them before we use them. But what are the advantages? And so this is sort of going through that. But I think the most important thing is that regulation is a dynamic process. It's not, regulators don't affect the accumulated amounts of their target genes. They affect the rates of synthesis and degradation, et cetera, et cetera. They affect the localization, the transport. They affect things that are all rate processes. There's nothing in chromatin structure and transcription that isn't something that should be, I think, at its core, thought of as a rate process. Because the only equilibrium or steady state cell is a dead cell. I don't know. That's a little rough. I don't think that's right. I guess, but even post-mitotic cells are doing lots and lots of stuff. So if we want to model the actual biology, we have to model the dynamic processes. We have lots and lots of things that will help us learn these networks. So we believe that these networks, given the scale or the number of components, and given a lot of theory about complex systems, there's a lot of reason to believe that these networks are sparse. And we can go into that more, but one of the key reasons is it comes from ecology, comes from this idea that connectance should scale as 1 over diversity, or connectance should scale over 1 over the component parts. This is sort of the May work early on. Whether or not that's true, we can see, but there's a lot of reasons to believe that sparsity is a good constraint. We know that one thing that helps us in biology is that we can set up cells, and then we can set up cells again, and we can set up cells again, and we can often get cells to be a complex system that's in a similar state. We can make many clones of those cells, and then we can do destructive measurements and stack them back together to make fake time series, to make longitudinal studies where the different time points are comparable. And think about trying to do that with Twitter or Facebook to study, if you want to study the destruction of Western democracy and you needed some Facebook data. This is being recorded, so hey everybody. You couldn't set up society 15 times and then do destructive measurements, like get rid of this server, deny these people access to Facebook. You couldn't do that and then repeat it. So if you do a set of destructive measurements in biology, you can do different destructive measurements and then stack them all together into a mega data set and try to learn something from it. We can't really do that in economics. We can't really do that in social networks. So we have opportunities from a complex system perspective that other scientists don't. The main thing is that we can do things like ChIP-seq and ATAC-seq and then combine that with things like RNA-seq, and we get structure constraints and state variables. We get to put them back together again and hope for the best. Now, not that that's easy, but we could do it. It could be done. Another thing I think that's important is that to make a more biophysical model, one of the key things is that we have to actually model activity as what it is, which is a concentration. So activity is the fraction of the thingies that are active. And so if this is a transcription factor, what does that mean? It's not an easy thing to think about. If it's STAT3, maybe the activity is the fraction of the STAT3 proteins that are phosphorylated at the right spot to be active. Do you then define activity as those STAT3 molecules that are phosphorylated and also in the nucleus? And do you then, oh God, it's crazy. All right, so I don't know how to do it exactly, but what we do is we do actually try to model this as a latent parameter in a model that has dynamics as its core generating model. And we try to do that with model selection being heavily influenced by orthogonal experiments like ChIP-seq and ATAC. So that's the whole sort of thing. Some of the rest of this is all a little bit more philosophical, but one other sort of thing that is a sort of mathematical idea at its core is when people look at different cell types in human, for example, they often learn different regulatory networks for different data sets. And those aren't just, there's different expression in the model, there's different state variables, there's actually different interactions. And so if the interactions are different, what does that mean? The sequence of the regulators didn't change. The cell didn't fundamentally change. It has the same genome and the same potential to express the same things, the same affinity of regulators for parts of the DNA. And what that means is that something else modified that regulator, a phosphorylase, a kinase, an interaction with another protein. And so interactions are perhaps the key to not having a different model for every cell type. But interactions are also a huge complication in terms of model size and model selection. So we're going to get to here, and we're not going to get to here. And all of what I just said is put together into a paper that I wrote with Tarmo Ajo, who was here actually a few weeks ago at talking about spatial transcriptomics. The two of us have put this together in a review that actually lays this out a lot more systematically than the last 10 minutes did. Yeah.
Just to clarify, do you mostly mean the activity of a protein and interactions between pairs of proteins?
We're interested in the activity of transcription factors. And the way that we define that activity today is its potential to influence the rate of transcription of its target genes, which is a great way of just taking everything and smushing it into one latent variable. Again, partly due to the limitations in the data. Okay, so again, another way to say this is big data if you're a social scientist. This is big data if the APSR survey is your main thing. This is big data if you're interested in human translation or vision. But in biology, we have much more complex situation where we have to integrate many different data types. Some give us information about structure, and some give us information about state. And this is the overall system that we'll talk about. Dayanne's going to talk really about this key step here, and I'm going to talk a little bit more about some of the details in terms of how we get these things. But in the end, we have state variables that come from RNA-seq. And this could be time series. This could be RNA-seq following knockouts. This could be single cell or spatial RNA-seq. We then turn that into differential expression and then use that as the main thing that we're trying to predict. The expression of the targets is the main variable that we're trying to predict. Good predictors of that expression will be edges in the network. One of the key advances in the last 5 years, there's been a huge amount of technological advancement in figuring out the structure of chromosomes and where things bind on chromosomes. And in the beginning, we often generated priors on the structure of network from ChIP-seq. And there, for each regulator you're interested in, you have to do an expensive experiment. And lately, there has been a lot of advancement that I think many people in the audience are aware of, where we can look at open regions in the chromosome and then cross-index those with databases of binding site models that have grown tremendously in the last 5 years, thanks to work by Tim Hughes and all kinds of other people like him. We can then get predicted binding for thousands of proteins. And so the first part of this talk, we'll talk a little bit about the core model we use to do network inference. Then I want to show you some of the difficulties in using this kind of work. So binding is not functional. And so when we use priors derived from real experiments, aka we're not in B subtilis, B subtleness is a great place to be. If we're in model systems, we might know thousands of interactions. If we're in humans or a mouse, we don't. And so we have to use these very noisy experiments. And so the key is to use this stuff that's only functional 1 out of 10 times if you're lucky to influence the model, so that you have a constraint on your model that isn't just sparsity, but it actually has some influence from these orthogonal experiments. That's what we want to build up to. All right, and so when you're going to do methods development, it's always great to have a Swiss German on your side, get some really solid data for a really easy system to work with. So this is Patrick Eichenberger, and he is he and I a few years ago built an array. This is all, again, ancient history, but there was a European effort that did things with a tiling array. And again, this organism is small enough that we have a gratuitous number of probes per gene, and so this is fairly high quality data. We've also done RNA-seq now as well, and we also have ChIP-seq, and the really key thing here is that we actually know 2,900 regulatory interactions for a genome that only has about 3,000 expressed genes. So for more than half of the genome, we actually have high quality, multiple experiment verified interactions. So the first step here that we want to pay attention to is, if we have these known interactions, can we back calculate the activity of a transcription factor? And so this is just the first part of this little flow chart. But again, who here would consider them more of a, who in the audience thinks of themselves as more a computationalist than an experimentalist? All right, everybody else get out. No, okay, so as computationists, we have to do a lot of things, but one of the things that we have to do is we have to convince our collaborators that it's occasionally better to estimate something than to try to measure it. A super noisy measurement can be worse than a good estimation. And this is one case here. So let's imagine we have one transcription factor like ROR gamma. It's this giant, you go to the PDB and look at a nuclear hormone receptor. It's this big mess, it looks like a clam, and in the middle it usually has like a ligand that's the size of cholesterol or bigger. Giant thing. Most of the nuclear hormone receptors are orphans, so they bind a ligand to become active, and we don't know what that ligand is. And that includes ROR gamma, one of the most important transcription factors for inflammation. We actually have some ideas, but they're all like top secret. They're being turned into drugs, so who knows, right? Many transcription factors, the leucine zipper, for example, most of the key transcription factors that were the first transcription factors that we learned about in the 50s, they all bind and form hetero and homodimers before they're active, and that's just the beginning. There's obviously a lot of oligomerization that influences activity, and then there's phosphorylation and modification of different kinds. There's localization. There's probably 50 different mechanisms that modulate the activity of transcription factors. If you want to get enough transcription factor activities for enough transcription factors, you might have to do hundreds of assays for thousands of time points. And so it's not really feasible to estimate activity, measure activity. We think that the best way to do things is to take an RNA-seq experiment of any kind and use the expression of target genes to back calculate the activity of the transcription factors. This is an old paper, but I think this is an interesting paper to look at for people who just want to scare themselves a little bit. So this is Sri Kusari, who was in the neighborhood and now is at UCLA. But what he did is he made all possible synonymous GFPs. So all of these, every little pixel here has the exact same protein sequence, but a different DNA sequence. So it's just like alternate use of codons and things. And what he found was that, first of all, the RNA, the expression of these genes, they all make the exact same protein. They all have the same exact promoter. The expression varied by a couple orders of magnitude. And in addition to that, when he then looked at the protein level, because it's GFP, so you can just sort cells, and there's a clever way of just using fluorescence to estimate protein concentration. Things that had the same, again, the same protein sequence had totally different relationships between their DNA or their RNA levels and their protein levels. So this is the ratio between estimated RNA and the effective protein level. So So in the end, there's lots of regulation that we don't understand, even if we just explore GFP. And so we believe that all the regulators, the activity of the transcription factor is the covariate that you care about. The expression of the target is the covariate you care about. So we can actually measure the expression of target genes. RNA-seq in all of its various forms is a very nicely mature technology. But we don't have the activity on the other end. And so one thing that we could do is we could just come up with a simple model. We could say the expression matrix that you collect is generated by the connectivity of the activators to the target genes. And so if this is our prior on the network, noisy as it might be, we can compute the activity of the transcription factors by applying the pseudo-inverse of the connectivity to the other side of the equation. So we can use essentially just a very simple linear additive model to compute activity. Now, should we? I don't know. Some of these things are repressors, some of these things are activators. But let's assume that this is a matrix that has -1s and 1s. -1s for repressors, 1s for activators. It turns out that if we just estimate activity with this simple equation, this is just 3 lines of MATLAB, this actually beats anything that we can test in B-Subtilis. Now, that doesn't mean that we're not actively trying to make this particular estimation better, but this is sort of what we get. So this is a regulator in B-Subtilis. So COMX is a quorum sensing molecule. So it's actually like a cytokine for bacteria, right? COMX is secreted at some steady state, and when population density gets above a certain level, it activates COMP, which phosphorylates COMA, and then COMA is regulated. Now, we know that this protein's activity is going to be pretty disconnected from its transcription level because one thing that varies a lot in experiments is the density of cells, how they were grown, what's been happened, how many times they were passaged. So we know that the activity of this thing is going to be all over the place, and when we actually look at our estimated activity versus expression, we see that there's a relationship, but it's a pretty wild relationship. And if we look at our estimated activity, you can see that it's just a reflection of the few known targets that we have for this. So this is very simple. And we can talk more about that, but what I want to do is actually just fit this into model selection, and then we can look at just empirically whether or not it works. So the next thing to do is to actually say if we have some estimated activities, so we take our guess at the network, we estimate some activities, and then we update the network. So we want this to run one iteration of model selection and see how well those networks are doing. Now, the ancient, the 2006 paper that sort of, where we first did this kind of stuff, had a lot of things that we don't think are important in there anymore, like our methods for modeling interactions and things. But this is the core model that still actually is a workhorse in the lab, which is that if we have some target gene or cluster of target genes, say Y, that the change in Y is some degradation, which we explicitly model, plus some linear or some squashed linear function of the regulators and their interactions. And within this framework, we have all kinds of variations that we've tried throughout the years, but this is a simple ODA model. The idea is we'd have one of these equations per target gene, and we'd try to learn the parameters for that full system. Okay, and we have in the past modeled interactions. We can do that with quadratic terms, but we've investigated all kinds of other things. And the simple way to do that is to say from the null model to the fully fit model, the early days we used cross-validation. Now we use Bayesian methods like what Dayanne will talk about. We also use stability selection. There's a million flavors of deciding how complex your model is, and they're all very, very closely comparable. But the idea is once you set the differential equation up in that way, you can approximate the gradient, you can put all of the steady state and the time series data together, and you can use the lasso, L1 shrinkage, the elastic net, the BIC, or any other of these essentially very comparable methods to learn the model. And to learn the model, we pick one shrinkage parameter per gene, and so we have a number of parameters in the model, free parameters, equal to the number of genes, and we have data, the number of data points times the number of genes data, so it's a doable thing. The best place to look at this all put together would be this molecular systems biology paper, which integrates the activity estimation and a fairly basic but still fairly robust model selection framework. And in the end, in that paper, we use BBSR. And here with BBSR, what we've done is we've used Zellner's G prior to put in a term G, which affects the balance between the prior and the likelihood in the computation of our model parameters, or the likelihood of our model parameters. And here the idea is that instead of using the Zellner's G prior with G as just a single number for your entire task, G is now something that is a vector that's the length of the number of regulators. And G is bigger if you're not in the prior and smaller. No, it's bigger if you're in the prior and smaller if you're not in the prior. And beta 0, beta naught, the prior, because we believe that these things should be sparse, beta naught is essentially a distribution that's tightly centered at 0. So beta naught is you're not a regulator, your value is 0, so beta naught is effectively 0, but it's really this distribution centered at 0. Beta OLS is the unconstrained model, and G is essentially saying that we're going to penalize things that are in the prior, in the ATAC-seq data, in the ChIP-seq data, in the gold standard less. Another way to do this would be to say that we have a penalty that is a constant times the sum of the absolute values plus a constant times the sum of the square of the model parameters, and that this should be some fraction of the fully fit model, or the fully overfit model, the unconstrained model. If we set this up like this, we can actually add another parameter here that is less than 1 for things that are in the prior, that is 1 for things that we don't know about, and if we had known negatives, if we had things that we knew weren't possible, or we had proper negative results, we can make this greater than 1 to penalize those things more. Yep.
The parameters in the prior are, when you say it's in the prior or not, are we talking about a prior on the relationship between?
That's right. It's not a prior on the value, it's a prior on whether or not there is a connection. And so the idea would be, in this first paper, what we did is we said, if you're, for example, in ChIP-seq, ChIP-seq says that there's a significant binding of this factor to the correct region for this target gene, right? That's I to J. If it's in the prior, we'd make this 10 to the minus 4. If you're not in the prior, you're 1. Something like that. And in this early paper, we found that these parameters didn't matter much. As long as you were something greater than 1, basically, you had a huge effect on model selection. If you were something less than 1, you had a huge effect on model selection, and there was a very large range of those parameters that mattered.
So the ChIP-seq data is giving you, it is mostly informing this prior on where we expect edges to possibly exist in the first place.
It only enters through these parameters, depending on what, and we'll mostly focus on this method for now. And
then the expression data is effectively the likelihood that you're
yeah.
Next one. Okay.
Yeah. And for anyone who bothers to read our papers, I mean, these are 2 of the ways that we use priors to influence model selection. But over the last couple of years, there's all kinds of flavors that have popped up. One of the reasons why all these flavors have popped up is that they're all pretty similar in their performance.
Can I ask a question about that? So are you saying this is how you incorporate like ChIP-seq data into your prior, or that you're talking about it as upweighting or downweighting a prior depending on the ChIP-seq data? I'm sorry, I'm a little
right. So let's imagine we use this. This is the simplest for the purposes of explaining the method, right? So we're saying that if you're in the prior, you're going to get penalized fractionally compared to if you're not in the prior.
So when you say if you're in the prior, if there's a relation, a TF gene pair for which you have ChIP-seq evidence, then we'll call that an entry of beta that's in the prior?
Yeah.
Okay.
And if we're sitting on one target gene and we're building a model for that target gene, if regulator I has a ChIP-seq hit to that gene, whatever that might be, if its motif is in an open region that's differentially open for that gene, if it's just in transfac or or the trust database. You know, there's a number of ways that you could be known or that you could have experimental evidence. So when it's the B subtilis or yeast, we have a mix of experimental derived priors and things that are pulled from the literature. When we're in human and mouse, we exclusively use experimentally derived components of the prior. So how you decide what this number should be is different. And how you take your experiment and decide which guys get a reduced penalty. The important thing here, though, is that we don't actually change the likelihood at all. We don't change the probability of beta given the data and the last iteration of the model. None of that gets changed. The only thing that gets changed is the penalty for being included in the model. So if you don't have a contribution to the likelihood, being in the ChIP-seq prior won't put you in the model. So most of the things, the important thing is that most of the things that are in these noisy priors are removed by the process of doing this network inference. And this is kind of what, this is from this B subtilis paper. And so we looked at Genie 3, which is from the neighborhood, CLR, which is kind of old but pretty robust, and the inferlator. The dotted lines are when we use expression of the transcription factor as the covariate. The solid lines are when we use the activity estimation from this extremely simple activity estimation procedure. And all of this is, all of these slides are for things that were left out of the training set and included in a proper evaluation set, et cetera, et cetera. So if we put these things together and use priors, we do much better. If we use activity, we do much better. And those two things together give us quite a bit of performance increase. The key thing here is that when we use activities, if we use these other methods that don't use priors at that second step to influence model selection, you see an improvement if you use the priors for model selection as well. Both stages, you see a bigger improvement. And the key thing here, there's two things that we're doing. The first thing is that we're coming up with a better covariate than the expression and showing that it's actually not that hard to estimate this, in this case, in B subtilis. We'll get back to human, where it becomes harder due to the complexity and all sorts of other things. But the other thing is, what is the prior doing when we go from this level of performance to that? And it's addressing the key problem in biological model identification of any kind, which is that there are lots of co-regulated genes. There are lots of things that form complexes. There's lots of block structure in any covariance matrix in biology. So breaking the ties between regulators that are under similar regulatory control themselves, that's the key to the game. So this network inference is not doing anything. Well, it's a fairly sophisticated way of breaking ties. And that's, I think, what we think this increase in performance is, really. OK, so when we put this all together, we get back to this method where stage 1, we estimate activities. And stage 2, we then learn the model. And some of this we'll go through again in a much more equation-oriented way when Dayanne takes the stage. OK, so I'm just going to take us through a little bit about what are the data requirements for this kind of stuff. In 2012, we put together a network based on lots of knockout, lots of ChIP-seq, and lots of RNA-seq. And we essentially only included things in the network if we knocked them out, if we ChIP-seq them, or both. And what that did is that limited us to about 60 transcription factors, because that was all we had in the budget for getting the antibodies and doing these things. Emily Moraldi and other people in the lab, including Dayanne, have found ways of generating priors now with ATAC-seq data. And so Olivia Wilkins published a paper in eLife where she did this with rice. Emily Moraldi, eLife and plant cell, and Emily actually has a paper up on bioRxiv right now doing this with Th17 cells. But the idea is instead of focusing on single transcription factors, we can use ATAC and motif databases, and we get noisier priors. But ChIP-seq's not all that magical to begin with, right? Because ChIP-seq is still binding, and you get thousands of peaks per transcription factor, and a small fraction of them are functional. But even if 1 in 20 peaks in ChIP-seq, or 1 in 25 motifs that are accessible or differentially accessible, even if a very small fraction of those are functional, it's still a huge reduction in the potential model space, right? Because it's 1 in 25 as opposed to 1 in 20,000. So there's still a manyfold. I think we're going to run out of time. So this is the key. This is where the key details are. So that B. subtilis paper shows the full pipeline applied to a model system. We really hammer out the details and work on the math. And this is actually a paper where Emily goes through and figures out how do we generate the prior in the first place. And obviously it's not that simple. A key reason it's not that simple isn't because of an increase in noise. It's not because human cells are bigger. The difference between a 4,000 component system and a 20,000 component system is not that big of a deal. I mean, there's basically a bacteria is so complex as to be mind-bogglingly screwed up. If you then go to a million cell, you're messed up, and then you're more messed up. But that's not the issue. The bigger issue is that they're paralogous. There's 7 stats. There's 50 nuclear hormone receptors. There's 60 AP1-like factors. The biggest problem with the mammals is that there's lots of redundant, very similar binding sites, and there's lots of co-expressed protein regulators that are in the same family. So the challenges for the model selection side of things are really complicated by the fact that there's a lot more redundancy and a lot more overlap in some of the things that we can use to identify these factors. So Emily and Maria collected a ton of ATAC-seq, and they collected a lot of RNA-seq, and they show that there are sort of interesting different ways in which the cell types cluster in these different data sets. So there are different things going on in the ATAC and the RNA-seq. In the end, this is something that we collected a few years ago. So this has been published in different ways, but the network is sort of always the last thing to publish. In the end, we have this ATAC-seq data following the polarization of these different cells. So we have knockouts. We have the time series as these cells establish fate. When we put that all together, we can use the ATAC-seq and the motifs to generate a prior and then put this into different flavors of model selection. And what Emily's paper, the reason why you might want to look at Emily's paper is because she really carefully looks at experimental validation of the activity. I'm going to skip that and instead, and she also makes figures that you need a magnifying glass and a half an hour to read, but the key thing here is that she tested endless combinations of methods for doing model selection, the strength of the prior, and the source of the prior. And I think that it's not that it's not interesting, but it's just tedious to test all those things. But one of the key things that she found is that many flavors that you wouldn't a priori know which flavor would be the better way of doing model selection were actually fairly similar. And so here we have pluses are the strength of the prior. No prior. Prior is so strong as to be absurdly strong. It doesn't allow things to be dropped. Something in between. We have stability selection and we have BBSR. Those are the main things. And Emily finds in this particular case that stability selection with the transcription factor activity and the use of a sort of intermediate strength prior is important. And these things kind of make sense in retrospect because if the problem is there's lots of redundant things, then perhaps stability and a weaker prior are important so that we can drop more of the prior. So this is somewhat consistent with there being a much weaker prior. But in the end, she learned networks for this giant compendium of many thousands of experiments and many tens of ATAC-seq experiments. She learned networks using all possible combinations of these various sources. So she used the trust data, so the literature. She used our ATAC data. She used it with the expression and the TFA, the activities. And then she also used the ENCODE DHS data for similar cell types. So she was able to evaluate these modern chromatin accessibility-based methods for building priors, and she was able to compare these. And in addition to showing that some performed better than others, this plot is showing that they're actually finding uniquely different edges between these different sources. And so I think this is a complex figure, but between this figure and that figure, I think we've made progress, but there's a lot to learn before we figure out the right way to generate priors for network structure. So we know it's a key component of solving this problem, but there's still a substantial amount of work to do. ATAC plus a half-complete motif database filled with lots of paralogous proteins that have essentially the same exact binding site. I think there's plenty of people out there that think that's the answer, but it's not the answer. We have a long way to go before we're done with that. That said, a lot of when we start to look at these hairballs and pull things out, The papers that we're writing now are filled with things that are validated in the laboratory, and there's lots of exciting edges that we found. We think that these networks, for the network here, we think that it has at the top many thousands of new regulatory edges that are likely to be correct. So there's a lot in there. I think I'm mostly out of time. But one key thing that we're interested in is now trying to take these networks and compare them across different parts of the tree. So this is a network that Emily's learned with Dan Littman's lab that's aimed at the innate lymphoid cell lineage. So these are the T cells that don't have T cell receptors. That's the sort of quick way of describing what those cells are. Another thing we're doing is also trying to look at the interactions between T cells and dendritic cells, specifically focused on the co-infection steps that are key for vaccine development in HIV. But it allows us to use our T cell networks and our dendritic cell networks in cooperation. And I think Emily's up, or Dayanne's up next. I think at this point I'll save time for a few questions. And I'll just say that, go to the important step. So this work was all done at the Flatiron Institute and the NYU Center for Data Science. And the Flatiron Institute is a new institute, 60 people that study biology, 60 who study astrophysics, 60 who study quantum mechanics applied to material science. Everyone is computational, and we're unfortunately not in this building, but we're in this building, so. But we still get to call ourselves the Flatiron Institute. The Center for Data Science is currently looking for people. We have 6 tenure track positions that we're trying to fill, so tell your friends who are on the job market. This has been a great place. I think the people I have to thank, Dayanne is one of them. Nick DeVoe is someone to thank here. Emily Moraldi, who was co-advised with Dan Littman, she's now at Cincinnati Children's. And there are a lot of other people. Carrie Lamb did a lot of early work with the model selection. And the other people on here mostly graduated. Tarmo and Dayanne, I guess you'll see Dayanne next, so I don't have to thank her too much. All right, that's it for now. Time for questions.
So I want to follow up on what Rich was talking about. And he focused mostly on when we have a big data set of gene expression data, how do we learn networks from it? But in this case, is the idea that I have multiple data sets. They are related somehow. And I want to learn model for each one of them, but somehow take advantage of all that data. So you already know that gene regulatory networks is what we're trying to learn is these edges between transcription factors and target genes and how they influence the expression of their targets. So I don't have to do a lot of that intro. We get a lot of gene expression data. We use our network inference algorithm, and we derive knowledge from it, which is these edges of TFs and targets, and we try to figure out which regulators are important for a particular process. As we know, the more data you have, the more power and the better your models will turn out. And so, and given the amount of data that's available out there for biological sequencing data especially, so here you can see that basically since 10 years ago, sequencing data is just growing and growing in the public domain. And so we ask, how can we get the most out of all of that data? The main challenge here, well, there's a few challenges, but one of them is the so-called batch effect, which is this technical variation that will explain how features are varying across the samples. So this is an example of, these were all control samples, but they clearly clustered into 2 groups. And the only difference between them is the date in which the samples were collected. And these are example genes that you can see that the expression is just varying across batches, and it follows exactly that design. So that's something that needs to be dealt with when you're integrating different data sets. In particular, when you're getting data from GEO, from the public domain. So one way is we can just not deal with it, which is usually what we've been doing. You get a data set, you learn a network from it. You get another data set, you learn a network from it, and then so on. The problem here is that you're not taking advantage of the data we have available, so you have a smaller sample size, and that's a problem. So what a lot of people have been doing is you put those data sets together with batch correct. And here, you would have the larger data set, but many of these tools do require some sort of balanced experimental design across the batches, and you want to have similar samples to make sure that the batch correction works, and also to not remove a lot of biological variability. And you end up losing any data set specific knowledge that you may recover otherwise. So what we decided to do is instead of integrating the data sets at the data level, make changes here so we can somehow transfer knowledge between the data sets and at the end of the day favor more similar models, but still allow independence, and you would still have a network for each of the different data sets. This is known as multitask learning as opposed to single task learning, which is what is usually done. And our hypothesis is that if we learn the networks this way, they will be more accurate because you're taking advantage of more data. So this overview is, again, I'm just going to repeat a little bit of what Rich said, but I'll try to be, just to set up the part that we're more interested in. So we have gene expression data. We have a prior network that he's already talked about. It's just these known interactions that we can get from many different sources. We estimate transcription factor activities, and then we use that as predictors to infer our network, which is basically as predictors of gene expression. So the first step is activity estimation. There are many reasons why we want to do that, because, and that was already talked about, but transcription factors, the mRNA level of transcription factors are not a good proxy for their activity because they need to be translated, and there's many interactions that are important for regulation. Also, there's many post-translational modifications that control the activity of those TFs. So we estimate that by using the prior network, which is the idea of looking at the targets of that TF to estimate activity. And the priors can be coming from different places. In this talk, we'll use one from chromatin accessibility data and DNA binding motifs, and one from literature. And then, yeah, so we use that partial knowledge of the network to estimate activities. And we already know it is, so in this case, it's an overdetermined system, so you have more equations than predictors, and so it's a linear fit. You guys been through this. So this is, again, that example of phosphorylated STAT3. Yeah.
Just because some people weren't able to come to the first talk of the series.
Oh, sure.
Just to make sure we're on the same page, can you explain the axes here?
Yeah. Yeah. So this is so the X matrix is gene expression, so genes by conditions, and that we know. The prior is genes by transcription factors, and if you have a known, so let's go back here. So if you have any evidence for that regulation between that transcription factor and a target, and that could be coming from many different places, maybe there is a ChIP-seq by that gene, or ChIP-seq peak, or maybe you did an overexpression and you saw that gene changed expression. So that's an experimental evidence that link that TF and gene. Then that would be an edge in this prior. And if it's 0, then you don't have any evidence. And then we're trying to estimate the activities. And so because we have way more genes and transcription factors, this is an overdetermined system, and we fit it by taking the pseudo-inverse of P, and this is just a linear fit, simple. Any questions?
Do you actually make the interactions you haven't seen exactly 0, or do you give them, some small potential?
So in the prior is 0, so it doesn't influence the activities, but later it could be included in the network.
And if there are some known negative ones, what would you do?
Negative, you mean repression?
Like they technically don't.
They definitely don't interact. So here, it doesn't matter, it will be 0. We could potentially do something in the model selection to not include that. I never ran into that case because it's really hard to define that something really doesn't regulate something in any condition of, yeah. So I haven't run into that situation. Okay, so when we estimate the activities, this is just one example for, it's a TH17 differentiation. And over time, we had a measurement of phosphorylated STAT3, which is in this case that the transcription factor is only going to the nucleus once it is phosphorylated. So it's not active, even if the mRNA levels are high, maybe it's just hanging out in the cytoplasm. So this is, and then here, this is a Western blot, so measurements of the protein that's phosphorylated, and this is a microarray gene expression levels, and this is the estimated activities, and our prior here was an intersection of a ChIP-seq knockout experiment. So when we estimate the activities, you see that the pattern is much more similar than the measured experimental data, but if we look at the expression, it doesn't look that way. All right, so that's the activity part, and now we're gonna focus on this part, and that's where we're gonna do things either in a single task way or in a multitask way. So now we have the gene and the gene expression and the levels of that gene in all the conditions, and we also have estimated activities for all transcription factors that we have priors for. And so we're trying to figure out which are the TFs that predict that gene's expression, and now in the matrix form, that would be the activities, which is conditions by transcription factors, times some unknown influence vector, and we're trying to predict the expression. So the difference between the single task and the multitask case is that you have either one data set or multiple. In the single task, you see that the weights that we're trying to figure out is just one vector. In the multitask, we have multiple of those vectors, and the best way of representing it is in a matrix like this. So each column is representing the influence of transcription factors to targets for a particular data set. So the axes are TFs by data sets, and here are the gene expression levels, and you can see that the data sets can have different number of conditions and things like that. Yes?
Is there any crosstalk between the different matrices? Because from here it looks just like 3 different
I'm gonna get there.
equations.
Yeah, I'm gonna get there. So our model is a simple linear model, and I'm just gonna stick with the steady state because Rich already talked about the time series. But the steady state model, it's just a linear model of so we're modeling the expression of the genes as a linear combination of the activities of the transcription factors. We want to find a sparse solution because we assume that only a few transcription factors regulate each gene. And so in this case, I'm using the lasso, which I think for most people here, you already know. It's used a lot, so that's one of the reasons why. And it's just the sum of the absolute values in this. So that will push a lot of the, so if each one of these is an interaction between a transcription factor and a gene and how much it influences, it will push a lot of them to 0, and you have a small number of predictors. This is the parameter, and we're using the extended Bayesian information criteria, which I'll get there when I talk about multitask. The other reason why we're using the lasso here is because when we uncouple the multitask way that I'm going to talk about next, it will be basically the lasso. That's why. So
go back. So what's white and orange?
What's what?
What does it mean to be white and orange in that vector?
Oh, white is 0, and then orange would be non-zero. So it's just, Yeah, so when you're penalizing for each, for the sum of the absolute values, that just means that they are independent, and then you send a lot of them to 0. And you want to find a balance between that and how well you're predicting the expression data.
Yeah. So you do consider the transcription factors to be independent of each other?
Yeah. Yeah, we're not modeling any interactions between TFs. Okay, so in the multitask case, we have the same model. They're still in. It's just to show that now we have multiple data sets. But if we look at this matrix, we can actually think about ideas of how can we transfer information between them to somehow make these weights more similar when they are supposed to. And this is for one gene. Okay. So if you think about a regulatory network, the main challenge here is that you wouldn't expect that the same network is active in all contexts. And we can easily think of a situation that in this one context, you have new interactions because that TF is active now. And in another one, this is a new TF that is, I don't know, now it can be expressed and it can work. So for example, if you have 2 cell types, then that's the master regulator of that cell type. And then the other cell type that's very close to that one will have a TF that will repress this fate. So that's just an example. So that poses a challenge of how do we actually model that we can transfer information and not make them only one single network. So I just want to, so if we sit at one gene, so this X gene, in this case, there's a few interactions that are shared. So TF3, TF7, they're shared interactions. But then for this data set, we have 2 other transcription factors that are active and regulating that gene here, but maybe not in the other data sets. And so if we look at the matrix, it would be something like this, right? So the TF7, all non-zeros, TF3, all non-zeros, and some of the other TFs will be just showing up in a few of the models. And just to remind you, each column is a data set, and these are the transcription factors, and then the orange boxes are the non-zero influence between that TF and this gene. So the way that we're optimizing this is by decomposing this matrix in 2 and regularizing them separately so they have this different structure. So the first one is one that's modeling kind of the independence between the data sets. So the transcription factors that are not shared. And then the second matrix is a block-sparse matrix that will model the shared components of the network. And again, this is for one gene. So now we're going to go through this slower. So what we're trying to find is the S and B, so these 2 matrices that minimize this function. The first part is just how we predict gene expression data. So gene expression for data set D, activities times the sum S and B, so the weights. And we want this to be low. And we will, at the end, sum over all data sets. And then a sparse penalty, which basically penalizes, it's a L1 both ways. So you're penalizing for each. It's the sum of the absolute values of basically every number in this matrix. So once you penalize for that, they will go away independently. And then the block penalty, which is, it's L1 L1 norm this way and an infinity norm this way. So basically, we take the maximum value. If you set in a row, you take the maximum value, and now you have a vector, and you can apply the lasso to it. So that's the penalty, and then that way you zero a whole row at once.
Is that what, that second matrix, you're enforcing that every row has exactly the same value, or that's sort of enforced through this optimization because of the L1 infinity?
So it doesn't enforce that they have the same value. They could even have opposite directions. They end up being very close to each other. But what we want here is just to make sure that once we zero a transcription factor, we zero it for all data sets. And then this matrix will actually balance even if you have like different influence values. Does that make sense? Yes?
So in B, you could have a row that has a mix of plus ones and minus ones.
Yeah.
But not typically.
In the same row.
But typically, yeah, you could. The optimization allows that because it's dealing with the absolute values. But typically, you wouldn't expect that to happen very often because an activator will likely be an activator in most conditions of that gene. There are cases that transcription factors will activate some genes and repress some other genes. I would imagine that there is cases that it will change. It will be repressing in some condition and activating in another, but I think that's going to be more rare.
But isn't that not what you want? Don't you want B to be a kind of common behavior across all your different data sets?
We care at this point about the structure. So if it's non-zero or zero, basically. And that way, we transfer information. So if you would have a weaker evidence for that interaction in both data sets, they will kind of supplement each other. And I'll give you a couple examples when we get a little bit later. Does that? All right, so now we need to choose the parameters, which here are 2. So for each gene, now we have these 2 free parameters that we need to fit. And first, I was doing this by cross-validation, and that just meant I have to wait a couple days or to do the network inference. So we decided to go with the extended Bayesian information criterion. And you probably are familiar with the BIC, which basically, so here is the likelihood in terms of residual sum of squares, and then you penalize by the number of TFs that you choose in the model, n is the number of samples. So when you add a TF transcription factor to the model, you're penalizing for that, and you need to find that balance. The problem is this breaks when you go to higher dimensions. And so people are starting to work with the extended BIC, and here is the citation, which basically scales with the number of predictors you have. So this is basically the number of predictors you have, choose the number of predictors you chose, and then it scales with that. It will scale with that. So just need some water. So we evaluate this in 2 organisms. The first one is one of our favorites, Bisodalus, that we had these 2 data sets. They were collected in different universities, so one of them at NYU and the other one in Germany. And then they also had different number of samples. They had a lot of overlapping samples, but a lot of them were also different. And we had a huge prior network that was literature-based, so very high confidence, about 3,000 interactions, explaining about half of the genes in the network. So it's a pretty complete prior. And the way that we evaluate is that we divided this in 2. So half of it, and we use as a prior. The other half we left out as a gold standard. And then using the prior and the 2 expression data sets, we did the network inference, both in a single task and a multitask way. And then we got our networks, and we evaluated with the other half that we left out. For yeast, we used 3 data sets. So in this case, they were more heterogeneous, I would say. The first one was the smallest, about 170 samples. All of them have a very different microarray platform as well, which is another challenge to integrating them in the same matrix. And this one was a chemostat compendium. This one is just ethanol, glucose, stress, and this one was more of like chemical and genetic perturbations. So they're very different conditions. But we still expect that there is a lot of overlap in terms of stress, transcription factors, and things like that. So we also had another difference here that we used a prior network that was not literature-based, because we wanted to simulate a scenario that you don't have that available. And here we used chromatin accessibility data. So wherever it's the genome is open, we looked for motifs, and we just assigned that to the closest gene. Very simple. If there were a couple of motifs, then this number would be 2. If there's no motif, this number is 0. And this is just the connectivity matrix between transcription factors and targets. And we use that again. So we use that as the prior. We had 3 expression matrices. We did the network inference, single task, multitask, evaluate with an independent gold standard. That was smaller than the one for Bisutulus. It had about 1,000 interactions. And there was an intersection of chip-seq, and knockout experiments. And if it had like 3 lines of evidence, then it was in the gold standard. So
one quick question.
Yeah.
So just to clarify, closest gene or closest promoter? Yeah.
The closest TSS.
Okay.
Yeah.
In the last study, previous slide, how are you splitting that prior network? Is it by nodes?
Random by edges.
By edges.
Yeah.
How do you normalize microarrays?
So I download them normalized. I don't have to because that's what I wanted to avoid, because a lot of times people, when they do their experiments, they know if they had to do any batch correction in that particular data set, they did. So I didn't want to go there, and I don't think any of these actually did any batch correction. But no, I didn't. The East data sets were all from SGD, so they've already done reprocessing of many East data sets. And these ones were actually were processed in-house by other people a few years ago. So I downloaded from their paper. Yeah. All right, so just to remind you, what we're trying to do is learn these 3 networks, or whatever many networks, and do some knowledge transfer between the data sets, so we have better networks at the end. And I want to give you an intuition of what knowledge transfer means here. So this is an interaction that is in the gold standard for Bisutulus. And so basically, we split the prior in half. We estimate activities. And this interaction was not in that half. And so when we estimate activities, and we're just correlating that estimated activities for this transcription factor and the target expression, this target. And you can see that for this data set, the relationship is very clear. For this one, you can imagine that when you bootstrap the data sets, you will see it sometimes, but you won't some other times. And these are the predicted confidence scores of that particular interaction when we learn it in a single task way and when we learn it in a multitask way. So the idea is that we do a lot of bootstraps, and this confidence score is related to how many times it will show up in the bootstraps. So in the single task one, we have a much lower confidence score for that interaction in this first data set. But when we do multitask, there is a transfer of knowledge from data set 2 to data set 1, and that particular interaction is boosted in terms of confidence score. For East, I picked this example, which was in the gold standard, and these are the 3 different data sets. This is the smallest one, and then it grows this way. And you can see there is a relationship between all of them, between the transcription factor and this target for all data sets. But when we learn it separately, the confidence scores are much lower than when we learn it together, in particular for the data set with the smaller number of samples. I think there is another way of thinking about it too, that this might be a way of breaking ties that you wouldn't know if you're learning from this data set only. You might have another transcription factor that's quite correlated, but it's not in this one, so you would have it transfer this way. So again, what we want to do is figure out whether this multitask approach is better than the single task one. So we're comparing each of the networks that we infer and figure out whether they're better by looking at the gold standard. And we do that by precision and recall curves. So just very briefly, precision is out of the interactions I predict how many are correct, and recall is out of the interactions that are in the gold standard, how many do I recover. And so I start at the top. That means that the first few interactions I'm predicting are correct, and as soon as I start going down, that means I'm making either new discoveries, so something that's not in the gold standard, we don't know about that interaction, or it's a mistake. So what we expect is that if the algorithm is better, you will have a higher area, bigger area under this curve. And so here, the dotted lines are single task using lasso, and the solid lines are multitask. And these are the area under the precision recall curve. So in both B-subtleist and East, we see that the multitask approach is doing better. In East, the boost is even higher when you look at the smaller data set, and that makes sense. Okay. So for now, I show you that when we learn these data sets together, the network recovery is better than if we learn it separately. And more importantly, we show that this is robust to prior types, so we use different types of priors, and we also use 2 model organisms and not only one, and we see the same kind of behavior. So now I want to talk a little bit about priors, and it's the same idea that yeah.
So how similar do the networks have to be for you to gain from multitask learning? Like if you share across, I don't know, like it seemed like these were supposed to be kind of identical and that's how you were sharing, but if you had things that you thought might be different, would you still gain by some small amount of sharedness?
Likely. So some of the experiments that I do want to do in the future is increase the difference. So maybe stay with the same data set and then get a very close one and like farther apart and see when that breaks. We don't have those experiments yet. It's also really hard in terms of you would have to really think about a good experimental design in that way. But it is a very valid question and something that it would be very interesting to pursue. I'm going to show you a little bit about like how is the overlap between the inferred edges. But yes, I would say that you will likely gain something if you have some amount of sharedness.
I guess I'd be curious in the human case about like different cell types, for example.
Yeah, that's where I want to go. Yeah.
Different cell types, and then also comparing human and mouse. Like how hard can you push this before it breaks?
Right.
Yeah.
I think that's future. Yeah.
Yeah.
Question?
Yeah.
So, related to that is whether the boost in precision recall is just due to better prediction of the edges that are the same versus also helping you on edges that are unique to one context. Do you have any sense of that?
What I can tell you is if I average the results, like at the end I have a multitask prediction and I average their confidence scores, then the performance is lower than if I take the maximum score for each, which means that the unique ones are also doing better. Yeah, that I can tell.
You said that to kind of have a gold standard, you have a prior where you basically, you divided it 50-50, you took out like half the edges and then kept those for testing. Do you think your results would be different if you did something more like 90-10 or like something where you're, in some sense I want to like take all but one edge and then test that one edge, and obviously computationally that's expensive if you want to do that over and over again, if it went out. Do you think it matters?
I think it will matter because the way that we infer transcription factor activities. So if you go very low, you won't have enough targets in your prior to estimate the activity as well. So we do experiments like you resample the prior, and then you estimate activity, resample the prior, estimate activity, and we see that if you have a very low number of targets, then that estimate will be less stable, which makes a lot of sense. So yes, I think there is a difference. It would probably hold if we do that, but I think that this is very realistic of a scenario that, so now I want to expand the B-Subtlest network, which is what was done in our lab last year, which was one of the papers that was cited before. And they used the whole prior, right, to expand that. And so if you take 50% and you are recovering the other 50%, that means you can do that to expand it. Yes?
Did you do some testing how large the influence of the prior actually was compared to just the fact that you're combining so many different data sets? Like what actually gives the gain in performance in the end?
So at this point, the prior was used only for activities, estimating activities. I didn't use it for anything else. And I think, yes, the activities is a huge gain, but I use activities for both, single task and multitask.
Maybe questions like comparing to rank combined batch effect. You're going to show that, right?
Yeah.
Okay.
Later. Did I answer your question?
I think so.
Okay. So now okay, let's go back. Okay, so now I want to talk a little bit about using the prior in the model selection step. So at this point, these are example interactions that I would keep that were in my prior, and I would be keeping in the inferred network in blue. And these red ones are, they're not there. But if I really believe my prior, then I want to make sure that those edges are kept, and I, make more discovery. But if I don't, then maybe it's fine. So we wanted to add a factor to fix that, and we added it to the S matrix. And it's a similar idea of what we were talking before, but here, so basically this number will be some number between 0 and 1 if you know that, if you have another evidence for that edge, which means I'm penalizing for that less. So, and that's user defined, so you can give a number, and then basically this will be 1 over that number. And what we wanted to see is, because we have very different types of priors, how increasing this number, actually decreasing this number, how does it affect the recovery of the prior, and how robust the method is to the different types of priors. Yes?
How did you choose to do that instead of like hard coding? Is it just so that you have like a continuous range of confidence?
Yeah.
So again, like in the first part, is it right that you were assuming that your prior was fully true to do your matrix factorization?
So in that part, we just assume the prior is true, and then we estimate the activities from that. And now we try to refine the network by, modeling the expression based on the activities that were estimated from the prior. So that's a way that the prior is already included in the model. But now it's the second step. So I'm trying to get the final output edges, right? And the idea is that if I really believe my prior, I want to make sure that the edges that were in the prior will also come out. Not make sure, but like at least push it a little bit more. And so this way, you could actually give different weights to different edges. So this edge, I have more evidence, and I would say it's a 2, and this one is less evidence, it's a 1. Not 1, it needs to be greater, like 1.2, and so this number will be 1 over 1.2 for the one edge and 1 over 2 for the other.
And so 0 would be, I don't want to penalize this at all because I'm pretty sure it's non-zero.
Right, I don't, that's a good way of, yeah, but I don't think I allow for that. Yeah, but that's something that can be done. I guess if you give like 100,000, then it would work, yeah. Yeah.
Should there be an interaction term between S and B itself? Because B is saying that one transcription factor is within the shared space, whereas S says it's a unique dataset specific term. So these are kind of mutually exclusive, because if one is predicted to be shared, it should not have the term in S to be like a lot. So you can basically get the vec of that multiplied by vec of this. That is to say, if something is predicted to be shared, like the elements on the rows of S should be all sparse. Right?
So I guess you could, but I just decided to let these parameters decide which one will weight more, and we'll use a separate measure to choose them. So I don't want to make assumptions, like more than I'm already making.
Yeah. The only thing I was saying is that there might be better if you have additional term that mimics the interaction between B and S itself directly, because this is independently like penalizes like sparsity in S, penalizes rows of B that are not full, but it doesn't mimic, it doesn't penalize the interactions between B and S that are contradictory with each other.
Right. And that could be good in a situation that the influence of a TF is important in all data sets, right? But in this one, that number, that activating number is twice as low, so you can fix it through S, for example. And that could be just something that is due to a batch effect. So like because the samples were collected in a different place, then I don't know, or the temperature, I don't know. It's something that's unmeasured. So I think that's one of the reasons why we decided to do it this way. But it is something to think about.
I mean, the explanations are competing, right, between sort of the B part explaining what we see and the S part explaining what we see, because we don't want to make both coefficients big, because that'll be penalized. So in loss, so we're kind of trading off which one we want to explain. So there's some implicit competition, I think, in the model, even if it's not modeled explicitly that way.
Yeah.
And related to this, for the NLP parameters that you end up with, you tend to have lambda b being bigger or lambda s being bigger.
So I have a grid, and I try them all. And for each gene, it will be different. There is a range, I don't remember by heart, that the original paper that started this modeling suggested, so I just used that. And basically, whatever lambda b is, lambda s will be between 1/2 that and the same, something like that. Yeah. Does that help?
Yes, because then they're penalizing b more, so that if there's something different, you'd want to put that in s because it's...
Yeah, exactly. Yeah. Cool. So we have different types of priors, and we wanted to know whether when we input that to the model and we increase the prior weight, if it will be robust to these noisier priors. So in this example, in the prior, the median number of TFs per target was about 13. So that's pretty high. And there were a few targets that had 40 transcription factors, and that was the motif prior, right? And you find way too many motifs around the gene. And then these are more conservative priors. This one was the subtlest one, was coming from the literature. And this is the East one that had that intersection of multiple experimental data sources. So when we do that, so here is predicted confidence scores for each interaction in the prior, and how, so basically if those are high, we're recovering those interactions. And in this direction, we're increasing the prior weight, so we're penalizing it less and less and less. And so for B subtilis, we see that those interactions are predicted with really high confidence, and that's our best prior, more complete. And then the East one, which is the intersection of multiple experimental sources of evidence, we have high confidence, but there is more variability than the B subtilis one. But when we use the chromatin accessibility priors, even if we put that to 10, which is a pretty high prior weight, we're increasing it, of course, because that's expected, but we're still robust enough to throw away a lot of the interactions because we do believe that this prior is much noisier. So this is good control. So we use the adaptive lasso to let the user increase the prior weight, but we're also robust to noise in the prior. And then finally, I want to have a look at the intersections. So as I said before, the first data set had a lot of shared conditions, and it's in particular on like sporulation of Bacillus, and these are more heterogeneous data sets. And we look at how the edges that we predict overlap for each of these organisms. And here is the Bacillus one. So about, and we're cutting off the network at about 50% precision if we draw that curve. So in the Bacillus one, we have a lot more overlap between the edges than when we look in the east, even if we had used different priors. So we use the gold standard as a prior, and then we get a network. And these are the inferred edges, how much they overlap. And if we use the ataxic prior, so the overlap here is much smaller, and that's really interesting because it shows that the model is able to adapt to more heterogeneous conditions. There is more to do here in terms of what we were talking about before, cell types and things like that in the future. But it is an evidence that we can do that. And then just to wrap up, we wanted to just double check with the other ways of integrating the data. At the end of the day here, we will only end up with one network. We are not having separate networks. So it defeats a little bit the purpose. But we wanted to compare if you do combine the 3 networks that came out of single task learning at the end of the day, if we do some batch correction, which here I use Combat because it's widely used and it's a good tool to join those 3 data sets that had no balanced experimental designs, and then learn a network from that. And then the other idea was that we can learn through multitask and then join them at the end of the day and look at these 3, how they perform, and doing the same things with the split of the gold standard. And for Bacillus, the difference is still huge. There is lower power to get those interactions using the lasso. And so red here is when we do multitask and combine. Blue is single task through batch correction, and yellow is single task and combine. And then this is for yeast. I think there is a lot more to be done here as well, especially when you have balanced experimental designs. And I don't think this is by any means a definite experiment, but it's really interesting that in both organisms, again, we're able to outperform in terms of,
How do you combine the 3 networks again?
I take the maximum confidence score of each because we wanted to still kind of keep the uniqueness around, So that's where I will end. So just to wrap up, we show multitask approach for learning networks from multiple data sets. We use an adaptive lasso penalty to increase prior confidence in the final model. Then we also show that this joint inference is robust to data set heterogeneity. And in this particular example, we can also show that MTL can outperform current integration methods. And that's on bioRxiv, so if people are interested and if you have any feedback, I also leave you my email in the next slide. No, not yet. So going back to what we were talking about before, the future directions of this is that we do want to start pushing for multiple cell types because we do believe that a lot of the interactions are shared, multi-species with the problem of figuring out orthology information and until what point you should actually join different species, and again, multiple conditions depending on So with that, I want to thank everyone for being here and listening. And for the questions, this was really, really fun. I also want to thank my lab. This was a fun bowling day in New York. Would like to thank my advisor, Rich, who's I mean, really funny. You guys saw. And I leave you my email. If you guys have any suggestions, I mean, you can come talk to me, and you can also email me.